So, oh, that's pretty loud. Okay. Tonight I have the pleasure of introducing Dr. Andres, who will be continuing NYSC's hidden goal of turning us all into neuroscientists. Dr. Andres majored in biotech, then pursued his PhD at Harvard. I could tell he had a sense of humor when he compared picking a graduate program to dating and getting married early on. He loves everything about airplanes, which he learns about from his brother, who is a pilot. In his free time, he also runs a science education program in Bolivia, through which he met our very own Fer. I actually talked to Fer about this, and she told me firsthand how much this program has impacted her, and how it's the reason she's here with us today. So I already have to thank Dr. Andres for inadvertently giving me an amazing bunkmate. Only connection to this camp. Dr. Andres was a delegate to the Youth Science Leadership Institute in 2006, which he described as a sort of pilot spin-off to the NYSC we all know and love. He was also a staff member in 2008, acting as the NatSci coordinator. I asked Dr. Andres what it was like being back to Camp Pocahontas after 11 years, to which he responded that it was like nothing has changed. Without further ado, Dr. Andres.
I have been here 3 times. This is my 3rd time. The 3rd time is always the most important one. Everyone knows that. But she forgot to mention I do love ice skating as well. So maybe one day we're going to ice skate. So I have been told that about half of the lectures so far have been on neuroscience. This will be the end of the talk, so thank you guys for coming. So I think all of you guys will agree by now. I guess we don't have a clicker anymore. Okay. Nope. Anyways, by now we will all agree that the brain is one of the most interesting, complex issues, or even I would say objects in the universe. Who here agrees with me when I said that the brain is the essence of what makes us human? Right? Good. Glad to see I don't have anyone who's strong feelings for liver, kidney, gallbladder, appendix. Seems like the National Science Camp is working on everyone. So, have you guys ever, who knows how many neurons the human brain has? What is the number? 10 to the 11. 85 billion. Very close, 86 billion. Yes. We add a billion every year to make it. So, basically, by the time you're born, the majority, I'll say about 99% of your brain is already made. All these 86 billion neurons have been made. They have come together, and they have made these brain circuits that are important, and they're going to be with you for the rest of your life. How cool is that? Yeah. So I first got to see my very first human brain when I was here in the National Science Camp. So a directed study that brought some human brains, and that was the coolest thing ever, and this is when I knew I wanted to study neuroscience. I don't have human brains to bring today. PSA is not very keen on letting me go with a human brain. But I do have something cooler I'll show you in a little bit. But of all of you neuroscientists, which part of the brain do you think, which one is your favorite part of the brain?
PFC.
PFC. What's the PFC? Prefrontal cortex. Well, someone said hippocampus. Amygdala. Okay, but you guys didn't explain why.
It's the logical center of the brain.
I'm afraid if I would have enough answer something else.
Corpus callosum. Sounds cool. To me, the most interesting part is the cerebral cortex, including the PFC.
Yeah!
Needless to say, everyone can have their own favorite part. There's a lot of brain. But I like the brain, the cerebral cortex, because it has some very interesting functions. Cerebral cortex, for instance, is in charge of creativity, language, memory, sensory perception, and very fine movement. I will make the argument that the cerebral cortex is the part of the human brain that makes us human. In fact, makes about 80% of the cerebral mass. It's the part that has the most dramatically expanded in evolution, and it consumes about 20% of your body's energy. So 20% of everything you eat goes to the cerebral cortex. How cool is that? And if you actually look at the cerebral cortex of across species, and by the way, the cerebral cortex is the outer part of the brain. It actually looks quite similar. So even though our brains have dramatically changed shapes throughout evolution, the cerebral cortex itself has not changed that much. It has expanded though. So my PhD advisor used to say this. We might all have different functions for our brain. Every species uses slightly different, every person uses slightly different. But the actual core parts and the brain connections are the same. In fact, if you actually zoom in into the cerebral cortex of our species, you will see that the same laminar identity and the same number composition of neurons is virtually the same. Okay? And if you actually zoom in even more into the cortex, you will see that it's made of two big kind of neurons, big classes of neurons, I'll say. These are the excitatory projection neurons, as their name says it, they excite the brain function. And they make about 80% of the neurons of the cortex. And there's also the inhibitory interneurons, which they all come in a lot of flavors. But the inhibitory interneurons, as their name mentioned it, they inhibit the cortex. And you actually need a very precise balance between the two of them. The moment you don't have a good balance between excitatory and inhibitory neurons, you basically, it's the fundamental mechanism behind most of neurological and neuromental disorders, including autism, schizophrenia, bipolar disorder, and more. Can anyone describe, for instance, autism? What do you expect to see? More or less excitatory neurons, more inhibitory neurons? What's going on?
More inhibitory?
More inhibitory. Why?
Because it seems like brain function is diminishing, so they prefer what's able.
It's more inhibited. One person is more quiet and tends to have more predator behavior. But you either have more number of interneurons or you have more inhibitory connections coming from a single interneuron. What do you guys think about schizophrenia? Excitatory, right? You make up things. You think about new things. So the moment you have more of one type of neuron versus another one, or less, that's when you have disease. So this is important, and this is a thing that our body has already figured out how this balance is made even before we are actually born. How cool is that? So in my group, what we study is we use this tool called single-cell RNA sequencing to actually create a map of the human brain across development. So who is familiar here with RNA sequencing? Okay. I don't know your name, but
Arnav.
Arnav, what is RNA sequencing?
Essentially, you're able to get the sequence of the certain RNA within that cell. And the really cool thing, I think, that we do with RNA-seq, I know it's here, but like we're able to tell to the sensitivity of RNA and the other specific parts of RNA, not only by like the sequence or like the specific shape or like morphology, I guess, of the code, but like also just like based on like the way we have to do it. So the person did it really nice and still like this.
Yes. Alex, Alexander, Alex, right?
Yeah, to add on to that, what makes RNA sequencing special compared to just plain DNA sequencing and sequencing the entire genome is that RNA sequencing really tells us which genes are being expressed in these cells. So, for example, for cells in the brain, if we use RNA sequencing, we can see which parts of the human genome are actually responsible for the brain.
Exactly. So, what we are saying basically, RNA sequencing is a tool that allows us to now understand which genes are being present in a particular tissue at a particular time. Make sense? Now, until very recently, this was done what we call bulk RNA sequencing, which means you take the whole tissue, you grind it up, and all of a sudden, you basically measure all the genes that are expressed in the human brain. That's cool. But if you think about it, it's like if you have smoothie of multiple fruits. You have multiple types of neurons, you have inhibitory cells, you have excitatory cells, you're blending them all together, and this is what you're spitting out. And this is cool in principle because it tells you what generically, on average, is being expressed in the cell, in the brain. But it actually doesn't give you a single cellular solution, like a fruit salad. Right? A fruit salad, you know, you have the same ingredients as the smoothie, but you know that you're eating an apple or a kiwi. Right? So single-cell RNA sequencing is now a form of doing the same analysis, but actually know the gene expression level of every single cell in that tissue. Make sense?
Yeah.
So that's what we do. And we have actually done it from 45 primary tissue coming from 45 donors that are human donors. And we actually have it across developmental stages, meaning we know during the period of neurogenesis, meaning the gestational week, second trimester gestational week. So we have 45 samples that we have sequenced the RNA of the human brain, the samples. And we have created to date the most complete map of gene expression in the human brain. And we can see basically, for instance, a bunch of things including inhibitory interneurons, as well as other excitatory neurons. And we have now basically a very nice report card of what every neuron in the body should be expressing at any particular time. And I'll give you an example with just 2 genes. And this is again on the cerebral cortex. This is a piece of the cerebral cortex of the human brain at gestation week 14 and gestation week 11. And you see these 2 genes here. One is marked with in green, it's called BCL11. B, and the other one is called SAPI2. And you can see, for instance, that in the prefrontal cortex, over here, these 2 genes are expressed virtually in most cells together. But if you go just a little bit farther to the visual cortex, these 2 genes do not localize. So here, basically, these 2 genes become a smoothie. Here, they become a fruit salad, almost actually a salt and pepper. They tend to actually be mutually exclusive from each other. So we have made this map for every single gene in the genome. So what can we do with this information? Anyone have any idea? Jake,
Target specific diseases.
you can target specific diseases.
I think you might be able to help like a map of certain functions in certain areas.
You can map certain functions in certain areas. Great. Any other ideas maybe in this side of the room? Sorry, I don't have your name tag. So. Oh yeah. You can recreate the human brain, for instance, in the dish. Right? Thank you, Alex. You got me exactly where I wanted to go. So we don't have to forget that we all started here as a single cell when that sperm met mom's egg and made the zygote. And we started a single cell, we very rapidly divided, made 2 cells, 4 cells, 8 cells, 16 cells. Eventually we made a little blastocyst. Here, this part became the whole body, this part became the placenta. Interesting in general. But what actually was at some point, we made and we were pluripotent stem cells. Does anyone know the definition of pluripotent stem cells? What is a pluripotent stem cell? Over there.
There's stem cells that can change in any stem cell type or any cell type.
So, there are cells that, as their name says, they're pluripotent, they can make a lot of things. And they can actually make every piece of tissue of the human body. Right? So, we can actually, in the lab, take this human pluripotent stem cell and culture them for long periods of time. Actually, you can culture them forever if you want to. And you can actually then tell these cells, I want you to become the pancreas, I want you to become neurons, I want you to become eye, whatever you want to make it. Isn't that cool? Yes. So this is how like the human pluripotent stem cells look like. This is a colony. Each one of these dots is a cell. They kind of tend to clump together, and we put them on top of some feeder cells that produce enough protein for them to eat. And then we can take these cells and push them to differentiate into any lineage of the body, including the brain. So, has anyone ever heard of the term induced pluripotent stem cells?
Yes.
What are induced pluripotent stem cells?
I've done this like 2 years ago, so I'm not certain. But like, essentially, is it like that you can then develop those stem cells? It's not like you find them, but you can take a cell and you can turn it into an induced or iPSC, and that you can then use like the pluripotent stem cells.
So we can actually, so what you're trying to say is that we can actually take any cell of the body and make them believe that they're a pluripotent stem cell. We can convert them or reprogram them into a pluripotent stem cell. And this is accomplished through a series of different methods, including the expression of 4 particular genes. But now we can basically take a skin sample from any of you and make your own pluripotent stem cells, which then we can differentiate into neurons, into cardiomyocytes, into I don't know, your God. And we can then, for instance, transplant it back to you if you really want it. So let's assume, for instance, that you have ALS and you don't make these corticospinal motor neurons, which are the neurons that connect your cerebral cortex with your spinal cord, and you cannot move. Well, we can take your skin cells, reprogram them into embryonic stem cells, or into pluripotent stem cells, sorry, and then tell them, I want you to become a corticospinal neuron. They will become corticospinal neurons, and you can transplant them into a patient. This is something that we can normally do in the lab, and we start usually, you can start with any tissue, but we prefer, most of the protocols prefer to start with skin fibroblasts because they're very easy to access. You literally take someone's arm, you make a little punch, and you have your cells. So, you start, you culture them, and then eventually you start pushing them to become induced pluripotent stem cells, and here by basically day 18 you can see that the comment resembles very similarly to what I showed you before that a normal human pluripotent stem cell will look like. So we are pretty close. I mean, we can do this from people with no diseases. We can make it with every disease you can think of. There's probably already an induced pluripotent stem cell. There's about 2,000 different lines around the world. But we can also make this little trick in which we can take about 5,000 of them and put them in these little plates that look like a V at the end. And what that does is actually it forces them, rather than become cells, they become now little spheres, right? Just put 5,000 of them and they tend to aggregate with each other. And once they aggregate with each other, they make this little ball of cells that originally they are pluripotent, but then you can also force them to become whatever they want to become. For instance, the very first project by Yoshiki Sasai, what he did is he forced them to become optic cup. So yeah, I'm going to show you a little video where you're actually going to see on top is the same exact video. So one of them is labeling the cells, and the other one is actually just showing you the cells. And you're going to see how in 3 dimension this tissue self-assembly without basically very little direction into what really closely resembles that. And this takes about 2 weeks. So yeah, you end up with like this little model that looks at the very end. So here it starts imaginating, and you can see here it starts creating something similar to this, like an eye, eventually. But it's super tiny. It's in the lab. So that's pretty cool. Can we, but I'm not an eye biologist, so I don't care. I'm here to talk to you about the human brain. So could we actually induce these cells to become brain-like structure? So this is about 4 millimeters big, and it's actually a human brain organoid, which is now the coolest new model organism that is actually not an organism. Because now it's a piece of 3-dimensional tissue that very closely resemble the parts and the cells that are present in the human brain. You don't believe me? Well, I can show you, for instance, the section of the human brain. Here are the neuronal progenitor cells in the cortex, and here are the differentiated neurons, and this is the brain organoid. Much on a smaller scale, a few millimeters big, but it's actually the same structure. They're super small. They're about 3 to 4 millimeters big. But you have them right here. In fact, you have them right here. Whoa, magic. they're 5 months old. We can keep them alive for about 4 years, no problem. And there is 2 different ones. One has a lot of cells and it's labeled 13234. That is interesting because it's actually not a human brain organoid, it's actually a chimpanzee brain organoid. It's one of the 4 induced pluripotent stem cell lines coming from chimpanzee fibroblasts. And then the other, the one that is going over here, is labeled H2126. It's even more special because it's one of 2,000 or so human-induced pluripotent stem cell lines, but it's actually me. So what you have there is actually my neurons. Whoa. And if you look deeply, you're going to see some little black spots in a few organs. Not in all. I'll say especially in the chimp one. Brad, can you confirm that you see some black spots? What do you guys think the black spots are? Bacteria? Contamination? Huh? Tumors, it says. Jacob. Any other guesses? I have tumors. Dead cells. Geez, thanks. So dead cells. If anyone has another guess?
Are the cells accidentally differentiating from another tissue?
The cells are accidentally differentiating in different tissue. They actually differentiate into eye. So you have a little eye looking at you. That is connected to a brain. And we actually know now that if you flash lights to it, it responds. These are things, but I mean, they are responsible. This is great for the early embryonic brain. So it emits electrical signals. And we actually know now that it responds to light and responds to different wavelengths of light at different wavelengths of frequency, different frequency, and you can actually train them to respond differently. So you can teach them how to respond.
These statues of things that are grown by cells potentially transplant someone who had, neurological damage to them in a certain section.
Potentially. Maybe I'll show it to you later.
What's the way that you induce the changes? Is it within the cell, do you add certain hormones, or is there a way that you're adding causing it to develop? The one that interests me is the chimpanzee and the human. Is there really that much difference in the brain? Are they completely different chemistry?
Can you hold your questions? Because I'm going to get to all of them.
Awesome.
Yeah.
Cool. Do you guys want a couple more minutes to keep passing it around, or do you want to keep going on the talk? I don't want to start. Sounds good. Glad to see you guys excited about this. Oh, I guess. So we did some single-cell RNA sequencing first because that's all we know how to do. And basically here is a few organoids that are, and each dot here represents a single cell. There's some clusters of cells. Here you're probably not going to know what they are because I don't have them labeled, and that was on purpose because I don't want my competitors to know. No, I'm kidding. Actually, I know exactly what they are. There's, for example, dividing cells, the neuronal progenitors, which we actually call radioglia, some intermediate progenitors, and some young neurons over here, and some more mature neurons over here. So these are cells that were born a lot earlier than these ones, and they have been different. So basically what you're actually seeing is a differentiation trajectory of human brain development. So you start from the dividing cells all the way to mature neurons, and you have present all of these cells in each one of these organoids. And I can show you some molecular markers here I can see that there is at least one person who likes molecular markers in chat.
Yeah.
Does anyone else like molecular markers? So for instance, you have KX67, which labels dividing cells. So I can show you that basically the group of dividing cells is over here. This is the gene. Each one of these, the red cells or orange cells, depending on who you ask, is the level of expression. So the more red it is the more of that gene you see in that particular cell. You can also look for neuronal progenitors in particular, which are labeled by this gene CLSPN. This one I can never pronounce, but it labels neuronal progenitors, mature neuronal progenitors, PPP1R17. See, I did it well. Or you can label neurons in general. You have a lot of neurons in these brain organoids. You can have some young neurons which are labeled by SAPI2, which are over here, or you can have some older neurons which are labeled all the way at the end. So I show you here basically the whole trajectory of brain development. Just as we saw in the primary human samples, we can actually see it in the brain organoids. So this is cool. Someone here was drilling me on it. It's like, oh, you have a chimp now. Why do you think a chimp is interesting?
Because it's ancestor of humans.
Right, so it's our ancestor. We can maybe actually can help us understand what makes a human brain special. Because I like to think when I sleep at night that my brain is special. Right? But right now it's very difficult, actually I would say impossible, to access cells from a developing chimpanzee. I don't know who of you have ever tried to deal with a chimpanzee by going after and trying to take a brain sample from them. It's not easy. But taking a blood sample, taking a skin sample, and reprogramming and making these brain organoids actually give us a window into otherwise virtually inaccessible species. In fact, it's predicted that about 60% of the primate species will probably disappear in the next 15 years. So we might actually have a way to bank at least the neuroscience part, or if not, the whole induced pluripotent stem cell lines and recover the species. In fact, you can actually recreate in these stem cells, if you know every difference, for instance, between the human and the Neanderthal, in theory, you could actually recreate these mutations in your favorite human line and make it become a Neanderthal. So it gives us now a window into something that we could never even think about doing before. Someone asked me, how do you actually do this? It's not that difficult. You literally take about 10,000 cells. You put them in a jar that has like a V bottom, so they have to group together. And then you induce them for about 18 days with different inhibitors to forbid it from going to a different identity. So you tell them don't become a gut, don't become a liver, don't become a kidney. I know some people here are very passionate about kidney, but I care about brain. So you inhibit basically everything else, and then you tell them, I want you to become the brain. And that's what it is. You end up with two almost identical looking cells, cell groups in this case, human chimpanzee, and you end up with some brain organoids that look very similar, or at least have the same phenotypes than their own species brain. And I can show you a little example if you actually compare it to the human brain. And we couldn't compare it to the chimpanzee brain because we couldn't access any, but actually the macaque brain, okay, another primate. You can see here a piece of human brain at the stage of week 16 and a half, and a brain organoid. You can see that at least some of the same cells are in the same order are still present, as well as in the case of the chimpanzee and macaque. So you can ask, for example, what is different between these two species. We found this gene called mTOR having downstream target, the whole pathway to be different between human and chimp, or human and other primates. So you can see here, for instance, PS6, which is a gene that is downstream of this part of this gene. mTOR is present almost exclusively. It's present in much more abundance in the human brain, but it's actually significantly reduced in the monkey brain. And we actually know now that if we start treating these we treat human tissue with mTOR inhibitors, we can actually get them to become very similar looking to the chimp brain. So it might be a mechanism how our brain, with a single modification in a single genetic pathway, has expanded so much and make it so different. But let's stop thinking about primates. Primates are boring. They're kind of like same as us. We can also start looking about our species, conservation of our species that are interesting. Elephants, I am currently trying to set something up with the Thai government to try to get some elephant skin cells and make some elephant ornaments. And this is interesting because the elephant has like a very large cerebellum, for instance. Not sure.
Whoa.
There goes power.
Okay, so thanks for coming, guys. Okay, third time the charm.
Everybody shine your phone lights. Oh yeah, Mothman. Where's Mothman?
So who knows good jokes? Anyone wants a microphone? Should we try again?
The whole camp keeps on turning on and off, so maybe give it a second.
Okay.
Anyone know any good knock-knock jokes?
Who's there?
Actually, do you want to take questions?
Sure, we can take questions.
Alright, I got one knock-knock joke.
Let's go say
What's the difference between a piano, a tuna, and a bucket of glue?
What?
You can tune a piano, but you can't piano a tuna.
What about the bucket of glue?
I knew you'd get stuck in that one.
Thank you.
Okay. Yes, it even works. So I was saying we're gonna go probably after the elephant. We're probably gonna go after the whale. And I really would like to go after the pink river dolphin. Has anyone heard about the pink river dolphin before? Isn't that cool? Apparently not everyone here were fascinated by it. But we can go actually by conservation. We can actually now take and bank induced pluripotent stem cells from different species. For instance, most of the elephants that are still around are related to each other very closely. So, or even in the hippos. So we can take the cells, make iPS cells and make induced pluripotent stem cells, reintroduce through CRISPR, which we're going to hear about in a couple of days, variability, and put it back into the environment. Isn't that cool? Yes. And people like me can study their brains, which, again, I don't think most of you would love to do a whole PhD on trying to catch whales for their brains. But I'll make an argument that going to about a single species is boring. Having a brain of a single species, why? This exists in nature, and maybe even go after nature. It's something that you might not even ever think about having in nature. Okay. Is anyone familiar with chimeras?
Yeah.
Yeah, what are the chimeras? Sorry, over here. Yeah, so it comes from Greek mythology, as you mentioned, and it's a mixture. It's a single organism that is basically three organisms combined into a single one. You have a lion, you have a goat, and you have a serpent or snake. You could, in fact, do this exact same thing in a dish, right? You could, for instance, take a piece of the mouse brain, for instance, its inhibitory interneurons, when they are still progenitors, and transplant it into a human brain organoid. Why do you guys think this is even interesting?
For example, I know that there are some animals that like genes that light up that are really like the jellyfish that are useful to have so you can see the brain.
So since to look at species differences, for instance, that's what Jacob's saying. Something that is interesting in one species that might not be interesting in another species. Any other comment? Over here.
Maybe like resistance to diseases?
Resistant to diseases. That's a very good point. I never thought about it. That's super cool. Over here.
You mentioned inhibition cells, so maybe there's like overexpression of something in humans and we're trying to inhibit it with the mouse neurons that are present in humans.
Exactly my point, though. There's one little difference between the brain. There's a couple, but there's one very important little difference between the brain organoid and the real brain, which is the brain organoid doesn't actually make inhibitory interneurons. So we're taking them from a different species and transplanting them into the human brain organoids because the human brain organoids on their own are not making them. And in theory, yes, we could take induced pluripotent stem cells, make them into neurons, and then transplant them into the organoids. The issue is that for a brain, a balanced brain to mature, it takes about 9 to 10 months for a human. In the mouse, it only takes 3 weeks. I want to finish fast. So if these interneurons are actually one of the drivers of maturation of the circuit, we could actually accelerate the maturation of the human brain by having some of these mouse neurons in the human brain organs. So taking a few, and by a few I mean 100,000, and then transplanted into a human brain organoid. So here is starting time 0, or I guess actually 1, because an hour after I transplanted them. And I'm putting a little X of where I transplanted the cell. The mouse, so what you see in white or grayish is the actual human brain organoid, and what you see in black is the mouse neuron. Okay? You can see that just as in the brain, these cells are going to migrate. So after 6 hours, you move. Standard processes after 12 hours, move a little bit more. Standard processes after 18 hours, and move even more. Then you move a little bit back and you actually move inside the brain organoid by 24 hours. If you do this and wait for about 2 to 3 weeks, you end up with an organoid in which the mouse cells are labeled in red and the human cells are labeled in blue. That actually looks a lot like a brain circuit. You have excitation and inhibition, you have it throughout the whole thing. Isn't that cool? But what does that actually mean? It means actually that you actually have a circuit in a dish. And I'll show you a video. And in this video, the organ is going to be light blue, and it's going to, every time a cell has activity, it's going to turn red. Okay? Ready? And I guess not. Uh-oh. Can you guys see a flicker or anything? Yeah, just kind of flicker off. You basically are seeing that the cells tend to shine, but they actually tend to shine together. I don't know if you see it. And when they shine together, they actually imitate what you expect to see in a normal human brain. So I'll show you, if we were not to do transplants, this is a brain organoid, and each of these cells in a cluster is labeled by a different color. And you're actually, what I'm showing you is the fluorescence of that cell over time, meaning how active that cell was over time. Okay, so this cell, for instance, was super active, and then it quieted down, and then these other cells started peaking later on. So very uncoordinated, right? If I transplant the neurons from the mouse, you start to see that every cell in that organoid starts to have activity at the same time. They have rhythms, just like what you expect to see in a mature human brain. So that's interesting. But someone here was even pushing me more on what will happen if you actually were to take these organoids or take this human cell and do the opposite experiment, not transplant mouse cells into the brain. That's boring. Let's transplant the organoid into a mouse. So some other groups have done it. We have done it too. So here what you have is actually the brain of a little adult mouse. Here, in green is labeled what the human brain organoid is. You can see that it actually incorporated very nicely into the mouse brain. So this mouse brain is a chimera brain as well, because it has a piece of a human brain and a piece of a mouse brain. You can actually see, for instance, that the human brain integrates and starts making neurons just like what you expect to see in a brain organoid regularly. I've shown that this is actually my data, showing the exact same thing. You take the organoid, you divide it again, and you just transplant the cells, and not the whole organoid. You do the exact same thing. They stay together. But the cells actually start maturing, and they start maturing at a rate that is very similar to human. You start to see the complexity of the human brain now inside the mouse brain. What does it actually mean? We have no idea. But it's something that we're always looking for people who are interested in studying. But what I've shown you today, and this is actually conclusions of the talk, is that we can actually model the brain, but we needed first to make an atlas. We needed to actually understand how every brain cell is made, what is different between the brain cells, and how we can actually use that now to recreate those models in a dish. And now we can, for instance, start thinking about making chimp, making human, making autistic brains, making brains from species that don't even exist. And I think that's the power of neuroscience, and this is why I think everyone here at the end of camp should become a neuroscientist. Without the question that this is the group that I'm very thankful to. Over here, sorry, I don't have your name, but
Jay Lee.
Jay Lee.
So we kind of discussed this with a different presenter who was studying AI. But when you're growing brain cells in a dish, when you're, I guess the end goal to construct a conscious brain. And if so, how would you treat that ethically?
That's a good question, because we notice now that once you make the transplant, these organoids start having a firing frequency of 60 hertz, which is what is called gamma oscillations, which is also the definition of consciousness. So it is a difficult question. It's a question that we don't understand even properly, even how we start. What we as a group are doing, we are not doing the transplants as much. We're actually just comparing the human to the chimp brain or to the other species to understand what makes the human brain special. But we also have a new grant now that allows us to connect these human brain organoids to a computer. And for the first time now, you have a brain circuit that is actually resembling a human brain circuit. Well, we graded a computer can learn how to do that, right? So we have a few molecules, including this molecule called OptoPatch, that allows us to now, with a microscope, virtually connect these organoids to a computer, teach the computer. In turn, I would love to, because these organoids are trainable, I would love to have a computer train an organoid. So in principle, you have one organoid training the other one through a computer, right? Which would be pretty cool. But yes, having an AI application of this would be super, I think, is a very feasible future direction. And it's a direction that we as a group are taking.
Would it be possible to train these brains that you make language, to use language?
I don't know.
Like, I don't know.
They don't have a little mouth yet.
language, I guess.
Yeah, they don't have a little mouth, right? So, if you were to define a language as a frequency of firing for a particular sound, then you can say, okay, every time you fire at this frequency, I will give you a reward. And get that this cell is, by definition, being rewarded frequently because it likes that language. That that's a language, right? But it's not a real language because there's no communication other than frequency. So I don't know. Depending on how you define it, I guess.
Do you have like this, like if you grow a brain, like can you train it like an artificial intelligence?
That's the goal. We were talking earlier today, right? And I was like, I would love to put it in a robot and have the robot move right behind a neuron, like fires, right? What I didn't show you is that a lot of these neurons are actually motor neurons that are made by the organoids. So you could, in fact, have a motor signal, which would be pretty cool. Okay, can I start over there and kind of move slowly over here?
So, when you train them, like, what is their reward? How do you actually do the training process?
It's usually an excitation with the right light that makes them feel good. It's very simple. Sorry, I don't have a more complex answer. It's actually that simple. There was a different question over here. I'll let you. Yeah.
So when you make these chimeras, do they have any noticeable function with the program brain?
Did they have any what?
Do they have any noticeable difference in their behavior or function?
Right. So this is the difference, right? It's right here. So these are the human cells, right? And we know that mouse cells connect to the cells. And here you basically see that each of these human cells is very uncoordinated by themselves. But when you have the mouse cells, actually, the mouse cells seems to be coordinating all the human cells.
Sorry, I meant in coordinates.
Sorry? Oh, when you transplant it into the animal. You know, no one has done the test to see whether they're smarter, for instance, right? But we do know that they do connect with the mouse brain. So the mouse brain itself, it's not like you have two independent brains here. You actually have a single thing. And because this particular implant was implanted in the visual cortex, which is responsible for looking at things, if you were to shine light or something to this mouse, you can actually record simulation in the human cell. So we know they become a single brain. Now, no one has done like a behavioral test to see whether this is a real, this animal is smarter. I can tell you a different group, which I see some people are being worried about consciousness, or why not, which I think is a very important question. But there's also other projects going on around that are a lot more sensitive. For instance, there's a recent group at Yale that reconnected a dead brain and revived it. And he had it alive for several days, right? And it fired and it was a brain. So this is very recent.
It was like an April.
Yeah.
It was like that.
So it's super cool what we can do. Consciousness is an issue. I think it's something that we need to think a lot about. What we think is that this brain resembles very early prenatal brains.
Why do the brains only grow so large, and do you perceive a way to make them grow larger? Why do they?
That is a great question. So why did it only get to like 4 or 5 millimeters? I think there's one that is like 7 or 8 in there. The reason is oxygenation. So as you can see, when we make a brain, we basically will make veins, right? But the human brain in your body has veins that feeds them oxygen. So we think that what happens is that they get to a certain point, and then all of a sudden they're not being fed with oxygen, and therefore they cannot grow anymore, right? Because they cannot deliver their nutrients everywhere. So we are working on different ways to create like artificial ventricles in 3D printing. to like grow the cells around it and make like a gigantic organoid. So there's some other people who said no one says that the human brain has to have that shape, right? Like the shape that we characterize as kind of like a balloon, it has nothing, there's no rule out there that tells us that this is the way the brain has to be, right? So there's this other group, for instance, that puts pressure on top of it, and what they make is a single sheet, like it's super thin, but it's a sheet pretty big of human brain organoids, right? But they say human brain in principle is flat, right? In fact, the cortex is flat. When you see wrinkle, it's because it's too big, and you actually start wrinkling it in order to fit, right? It's physics issue. But yeah, so we are working on bioengineering approaches, but our groups are just flatting it and saying, it's still a human.
What's the estimate for the number of neurons out there in the world?
In these organoids, so the question is how many neurons are in these organoids? So that's about a million. Sorry, over there. I'll promise I'll come here.
Just two quick technique questions. How do you, can you explain a little bit about how you sort cells individually for like the RNA sequencing? And then could you also tell me whether or not you have to like destroy the immune system or breed for immune deficient lab mice so that you can put a piece of human tissue in them?
That is a great question. So I'll answer the second one first. So the question is, why doesn't the mouse brain reject the human tissue? There are special humanized mouse lines in which the blood of the mouse has been replaced by human blood, and that allows them to have a human immune system. And Leo can tell you all about it because he works with those quite a bit. Maybe during his presentation, you guys can interrupt him as well, or sign up for his directed study. But yes, we use immunocompromised mice. We use humanized mice. There's also immunocompromised mice, basically, as an alternative. The other question is, how do you actually sort? You actually don't sort, you do it unbiasedly. So you take a piece of tissue, you dissociate it, and you basically make a soup of cells. And then you sequence as much of the soup as possible at a single cell resolution. You actually put a barcode into each cell. And you then, based on the previous knowledge of tissue level, we can reconstruct back to what it used to be, and we can validate it with other techniques to be like, this is the right order and this is the right age, right? But yeah, so you do it unbiased.
If you had some viable cells from an animal that was already extinct, could you use those to study brains even though it was already extinct?
Yeah, so the question is, can you revive? If you were to have some viable cells that are frozen in Siberia from the mammoth, for instance, could you make an iPS cell and revive and make a brain organoid? The answer is yes. In fact, there's a group trying to do the mammoth. Right? And there's also some differences between the mammoth is very similar to the elephant, so they're basically trying to engineer the elephant cells to have every mutation that is different between the mammoth and the elephant. And then not need all cells that are in Siberia that might or might not work, but they actually just engineer it with genetic engineering. We can learn more about it as well. Okay.
Is it possible to take the organoid cells further and create an entire human clone in the
I
future?
couldn't understand the question. It's a question, take the...
Organoid cells further and create an entire human body, like a clone in the future?
Could you make a human body out of iPS cells? In principle, you could. In practice, you will have to be a very good assembler. Like, you need basically a really good engineer. So we do have, for instance, organoids that are, this is a cortical organoid, but we have thalamic organoids, and we can fuse them. And we can show that the connections to the thalamus come to the human brain, to the cortex, and the cortex goes to the thalamus, just like you would expect to see in the brain. So in theory, it's possible. In practice, you really need a good engineer. Alright, sorry, Rachel first.
Okay, so if you're looking for these stem cells to form into brain cells, do you have any theories behind the eyes forming from the chimpanzee?
The question is, why are you getting sometimes eyes and in your organoids? The answer is because the mechanism, the molecular pinning of how to make an eye is very similar to how to make a brain, because the eye is still part of the nervous system. So it's off target, yes, but in reality it's virtually the same process with different concentration of the same molecules. Alexander.
Hey, did you see, so, with the organoid cells that were implanted, human organoids, implanted into the mice, did you see any results of how effective they were, let's say, in solving mazes? Did you see any intellectual increases?
Yes, so the question is, are the chimeric mice that have a human brain in it smarter? The answer is we don't know, we haven't tested. Jenny. Sorry, I feel promised to go this way. So Jen.
So if you were to like completely create some kind of brain, like no matter what animal, would it be completely blank or would it have like some knowledge in it?
So the question is, do the stem cells have memory of what they used to be? There is such thing as epigenetic memory, and it's defined basically as some kind of molecular mark that makes these cells try to become a tissue in particular, and you can force it to become a different tissue, but it never actually becomes perfectly that tissue because it likes to become, I don't know, pancreas. And it happens sometimes. There are some ways to erase that through DNA demethylation or histone deacetylation. So it's doable to erase that memory. But you do see it every now and then. Like we have, for instance, an organoid that every time you try to make cortex, it makes hippocampus. And we cannot really explain why or genetic memory is there, but like, so there is some kind of a genetic memory, but it's of the tissue of origin. It's not the memory of the individual. If that makes any sense. Sorry, just to clarify, the core memory is placed through walls here.
Is there any like a clinical reasoning for why you want to make a chimeric brain? Like maybe I guess this is
So the question is, why would you ever want to make a chimeric brain? Kind of sounds like a, you know, like, sounds like a dumb project to be someone. I think so the mouse brain is it matures in about 3 weeks. The human brain matures in about 10 months. So if you can take which subset of cells are the ones that mature in the human brain or in any brain and make the maturation go faster and have an actual circuit faster, you could in fact accelerate the human brain maturation, right? This is why we went after that question, saying, okay, we think the interneurons are the master regulators of maturation. So if we take the mouse interneurons and we transplant them in the human brain, maybe in 3 to 5 weeks we can actually get a mature circuit. This is exactly what we got, right? Some other reason is to actually understand what makes the human brain special, right? If you can transplant mouse cells and still get some behavior in the organoid that if you were to transplant more than human cells, that means that those rules were conserved in evolution. But if you get a different behavior, you might actually start discovering some rules that make the human brain different, because it's not likely to accept the same rules, even if you transplant equivalent cell type from a different organism. Does that make sense?
Two quick questions. The first one is, do you see, I guess it's related to what we're talking about, do you see development technology between, I guess from like IVF and basically just normal development of like a brain and one that you're doing in a test tube with iPS cells? And the second question is a little bit, it's like very theoretical, and it's kind of like, can we use this technology to create super soldiers? As in like, you take like the fear, like the, like, I don't know, like the anti-fear part of a brain, and you put it into like a human, like is that like a theoretical possibility of this technology?
So the first question is, do you see any difference between the organoid and a real brain, right? The answer is there's some differences. For instance, the organoids tend to have more stress. The cells look more stressed in general. ER stress and glycolysis, which are 2 markers of the cells, probably because of lack of oxygen, right? Which we're trying to solve. And can you use this to make a superhuman? Probably not. but I probably we're not going to go into that direction. In theory, you can think a lot of different kind of questions that would be interesting. I'll encourage you to think about questions about like solving disease and solving evolution. But I guess pushing the next step in evolution might be interesting. You had a question, sorry.
Are human brain organoids compatible with any other animal except for us?
Sorry?
Are human brain organoids compatible with any other animal except for us?
Are they comparable to any other organism inside? Are they compatible? Except for mice? In theory, into any any mammalian organism, right? If you put it, it will integrate. In practice, doing it in a mouse is a lot cheaper than doing it in an elephant. Sorry.
So imagine you have like a full-size chimeric brain that has neurons for like a plethora of species. Would you expect the cells of some species to kind of dominate and direct the rest of them?
We are actually doing that experiment. We call it the Common Garden experiment, or the Noah's Ark experiment, where you have like a bunch of species and you mix them together and make a single organoid of like everything in there. We don't actually know the answer to that. In theory, you have some cells that divide faster. For instance, the chimp cells will tend to divide a little bit faster than the human brain, and the macaque cells will probably divide faster because you have a shorter developmental time in utero than human. So it is a possibility that one will overtake the other one. We don't know the answer to it yet. And we don't even know how these organoids will function, right? Sure.
Yes. You spoke about cells can be used to stem cells. And stem cells are like really like cure and all that. So
Right.
this is issues like does it help in like medicine?
Does it matter what?
Does it help in medicine? Like
Right.
diseases?
So the question is this useful? Basically. Yes, I think the reason why this is useful is because it's actually giving us a little window on how the human brain actually works. We can't fix something that we don't understand how it works, right? The moment, and so far if you actually think about it, most of the neuroscience and neurology drugs were discovered either by accident or as a good guess. But it was based on very limited information because your information is either a mouse or some other animal or a post-mortem sample. Or sometimes you get live tissue from someone who like goes and get a biopsy or something. But most people who go to get biopsy don't go for fun. I mean, not like you do on a date, it's like let's go to the doctor and get a biopsy of our brain, right? Like you do it because you have a disease and they want to use it to diagnose it. So those tissues are probably not completely normal, right? So the reality is that in order for us to even start thinking about curing human disease, we need to start working with human tissue. And very recently, and I'm talking about 2014 when this protocol first came out, this wasn't even free, right? And now, 5 years in research is a very small amount of time. So the possibilities are, I think, endless as the more people start getting interested in this topic. And I highly encourage you guys, that you're going to undergrad in a month, to really consider this kind of work also. So you have organoids, we need a lot of computational people. Computational biology, bioinformaticians, someone else telling me that we're going to do neuroinformatics, I think that's few, right? We need these people, we need to understand the human brain in order to cure the human brain. Just because I mentioned him, I'm going to let him ask a question and then I'm going to move over here.
So neural networks can get trained on only one task, while brains can usually learn many skills, and all those skills help each other. So if you were to take one of these brains and connect it to a neural network or computer, would you be able to allow it to learn many skills, and if so, how would you do that?
So this question is, could you actually get a single organoid to work like a brain and learn many skills at the same time, rather than what in AI we call neural networks, which is usually tasked to a single task, correct? In theory, yes. in practice, I still don't know what behavior actually is in an organoid that is kind of like floating in a dish as you guys saw it. So we're still trying to figure out how we actually assess what behavior actually means in these things. So any input will be very valuable. I think what will be very interesting is if a single organoid has multiple circuits going on there. And then we can actually define each circuit function as a single task. And then show that consistently this task is responded by this cells versus this task is responded by this cell. This might actually be the case within the brain. There is a little bit of evidence that this might actually happen, that intermingled cells might actually be part of different circuits. So we don't know. I think it's still too early to know. Back here.
I'm going to kind of follow up this question into more, I guess, biological computing. Do you know of people who have either taken a sample of tissue of brain or using them to some tissues and connecting not only inputs but also outputs into like some kind of simulated world type thing in order to like help that learn and then actually see like outputs and
So the question is, can you connect them to something that gives you an output, correct? So what we do is we connect it to these things called MEAs, or multi-electrode arrays, which is, as the name implies, it's an array of multiple electrodes, about 1,000 electrodes or 2,000 electrodes. And the output there is which electrode gets turned on at a particular time. Right? That's the best we can do right now.
So it's never been like creating a system that kind of associated environment on the computer?
Not yet. Not yet. We will do it soon though.
Okay.
Okay.
So if you consider like the lion and the tiger creating a liger, would you think that the survivability of putting a human with a chimpanzee brain would be more survivable compared to a human like a mice brain?
I don't know, but we have a chinobo, which is like when a chimp prints with a bonobo, which actually happens in nature. We actually have an organoid right now going on about, I mean, we have a couple hundred organoids that are chinobo organoids. So maybe in a year I'll let you know.
So, two questions. First, have any of your organoids ever rejected a connection to an MEA? And also, have you ever tried to connect any of your organoids to the vertebrate nodes or mineral section of mice or
So you have two very complex questions. So the one is, do you ever have useless organoids that just don't produce any signal? The answer is yes. Sometimes organoids are silent for some reason. Maybe they're shy. And they just don't emit any signal at all. And we don't know why, but it happens in about 10% of them. And the other one is, have you ever connected it to an output? We have it, a different group in London has. And what they did is they connected it to a mouse spinal cord and a mouse muscle. And what they showed is that they can get, when they excite the human organoid, they can get the muscle to twitch.
So since you technically like integrate human organoids into the mice or animals or like those things, can you also induce like human disease in them instead of accidental medicine?
Yes. In fact, a different group, also in London, has done something similar. The question is, can you study disease? Someone else has done it for Down syndrome, for instance, and has shown that the cells in Down syndrome behave a little bit different than the wild type cells. It's still at the beginning of days. People are working on this. We're working on autism particularly. So it's still very early on, but it is definitely something, a direction the whole field is taking.
I have two questions. So the first one is, you mentioned that you scientists are able to bring the brain back to medical abilities. Would that happen in some memories and feelings because the brain was dead? And secondly, are there any differences between induced pluripotent stem cells and pluripotent stem cells like in anatomy or
So the question is, so I'll answer the first question first. So the question is there a difference between naive stem cell, like in naive pluripotent stem cells or induced pluripotent stem cells? That was a debate in the field for about 10 years. People were going back and forth on this, and some people were very strongly agreeing that there is differences, and there are some people who said there's no differences. And I think at this point the agreement is that there's a lot of variability between both groups, but it's a lot more variability between these two groups than between these two groups. So the answer is a little bit more complex, but saying between the standard deviation of the normal, we expect them to be fairly equivalent. The second question is, does the pig brain that was revived have any memory? The answer is we don't know. And that is actually a whole ethical dilemma going on right now, because for all we know, sensory deprivation is what this pig brain had, right? Like, it was alive, but it could not hear, it could not listen, like it could not see, it could not feel, it could not smell, it could not, right? Right? So, for all we know, it was a complete sensory deprivation, which could be, if you think about it, a form of torture, right? But we don't know. I mean, the group took some extra measurements to keep the consciousness of it off during the entire experiment. But the reality is that if we were not to put those molecules in the media, we don't know what will happen. So that's why we have to be. So neuroscience, I think, is pretty cool, but we're also playing with the essence, I think, of what needs to be given, right? I think most of us, if I tell you, like, physically tell me, you believe you have a soul and you can physically point where in your body will your soul be, I think most people would point to their brain, probably, or they will point to your consciousness, to your brain, and you're playing with that, right? So I think it's something that you have to take with a grain of salt, and you have to be very passionate about it. I think the potentials of neuroscience work is incredible, but I think you have to be very conscious about how much you can actually play. It's pretty fun, but you have to be careful. Any more questions? I know I'm stopping you from your snacks. I think it's all done.